Okay, let's dive right into this explainer covering an absolutely fascinating recent paper from the ISME journal. Led by Michael Hoffert alongside Noah Fuhrer and a brilliant team out of the University of Colorado Boulder, this research tackles what is literally one of the most overwhelming challenges in modern biology. How on earth do we make sense of the microscopic world? I mean, just to put this scale into perspective for you, a single pinch of dirt from your backyard, it holds more bacteria than there are people on our entire planet. It's mind-boggling. So how exactly do we organize that kind of microscopic chaos? Well, today we're going to explore exactly how this team took an immense, wildly chaotic data set of bacterial genomes and transformed it into a brilliantly structured map. So, here is the core question that really sparked this whole project. How do you organize life that you can't even see? You see, right now, science is discovering new bacterial taxa, basically brand new branches on the microbial family tree, way faster than we can actually figure out their traits in a lab. And that leaves researchers stuck with this massive, chaotic biological data dump. We know for a fact these organisms are out there because, well, we have their DNA, right? But beyond that, we often have absolutely zero clue what they actually do, where they thrive, or how they're interacting with their environment. Which brings us to the exact problem, but also the inspiration for an ingenious solution. We all know Mendeleev's periodic table of elements, right? Think back to chemistry class. It's neat, it is perfectly predictable, and it organizes elements beautifully by their shared chemical properties. You look down a single column, and boom, you instantly know how all those elements are going to behave. But bacterial diversity? Yeah, it's the exact opposite of that. It is a chaotic, completely unmapped wilderness filled with continuous, incredibly complex traits. So the researchers posed a really bold hypothesis. Could we build a similarly functional, elegant map for literally the messiest, most complex organisms on Earth? Could we actually pull off a periodic table of bacteria? To solve what's essentially a massive biological cold case, this team didn't just look at a handful of common species. No, they went big. They mined a truly staggering dataset of 50,745 bacterial genomes. That is a huge number. It represents an incredible swath of the entire bacterial tree of life, pulled from 31 different phyla. And phyla are basically those major overarching branches of the family tree. By tackling this at such a massive scale, they made absolutely sure that whatever framework they built wouldn't just be some niche, overly specific tool, but a truly comprehensive map of global microbial diversity. But here is the kicker. How exactly do you figure out what over 50,000 different bacteria actually do without growing them all in a lab? Which, by the way, is physically impossible for most of them anyway. Well, instead of trying to measure everything manually, they used some seriously clever computer algorithms to just read the DNA and predict 6 fundamental traits directly from the genetic code itself. We're talking oxygen tolerance, whether they make their own food, if they use light for energy, how fast they can grow, their DNA's GC content, and their total genome size. Think of these 6 things as the bacterial equivalent to an atom's chemical properties. By inferring these traits directly from the DNA, they totally bypassed the slow, traditional bottlenecks of microbiology. Now just take a second and look at the sheer, overwhelming complexity of the original bacterial family tree. I mean, yes, it's an absolute marvel of evolutionary mapping, but trying to actually read it, it's like trying to navigate your way through a new city using a map that insists on showing you every single blade of grass. It's too much. If you tried to map those 6 specific traits onto every single tiny branch of this massive tree, you'd just end up with an unreadable mess of data. You literally wouldn't be able to see the forest for the trees. So to build a usable table, the team desperately needed a way to simplify this chaotic structure, but without losing the really critical big picture information. To finally find the signal hidden in all that noise, the team deployed an incredibly innovative mathematical trick. It's called a Haar-like wavelet projection. Now, don't let the jargon scare you off. Just think of it like taking a fine-tooth comb and running it through really badly tangled hair. The algorithm systematically travels down the incredibly complex branches of the evolutionary tree and basically smooths things out. It's hunting for areas where those bacterial traits are perfectly uniform. It pinpoints the exact evolutionary splits where a major trait change actually occurred, which highlights the branches where all the descending bacteria share the exact same functional lifestyle. And once that algorithm identifies those perfectly uniform branches, the researchers do something brilliant. They essentially prune the tree. They visually collapse all those messy, chaotic, individual tips into clean, unified groups. It's all about cutting away the unnecessary visual noise. And by doing that, these newly pruned, smoothed out branches can now act as the individual cells in our brand new table. It really is the perfect balance, you know? It preserves the deep evolutionary history of the organisms while finally giving us a functional map that a human being can actually read and make sense of. So, spoiler alert, they actually pulled it off. They created exactly what they set out to build, a periodic table of bacterial taxa, or PTBT for short. What we have here is a concise, highly visual framework that organizes these bacterial groups based entirely on the distributions of key traits that were predicted straight from their genomic data. This is a massive paradigm shift. They've moved us away from just grouping organisms by their somewhat arbitrary taxonomic names, and instead, we are now grouping them by what they actually do in the real world, their real, uniform, functional traits. And here it is, the final framework. Just look at how those overlapping traits, things like oxygen tolerance, overall genome size, and growth rate, align so perfectly across the board to define these major, distinct ecological strategies across the entire tree of life. You can clearly see these distinct clusters of bacteria that have evolved similar life strategies, all perfectly grouped together into nice, visually distinct cells. The dots and the bars just line up across completely different phyla. It's visual proof that specific, predictable combinations of traits recur over and over throughout evolutionary history, creating these neat, predictable columns, literally exactly like the periodic table of elements we know so well. So why does all of this matter? Well, it matters immensely. As the authors themselves put it, this whole endeavor is really about identifying clusters of co-occurring traits that define distinct ecological strategies. Think of it as a bridge. It perfectly bridges the massive gap between highly complex microscopic biology and massive real-world ecosystems. By actually knowing the functional traits of these bacterial groups, we can start to accurately predict how these invisible communities operate out in the wild. Whether they're cycling carbon deep in the soil, surviving in the most extreme environments on Earth, or literally interacting right now inside the human gut. As we wrap up this explainer, I really want to leave you with a final thought to chew on. What untold mysteries, what bizarre and unique ecological strategies are still hiding out there in the uncharted branches of this incredible new map? Remember, we have only just scratched the surface here by looking at merely 6 traits. I highly encourage you to dive into the full paper and explore this map for yourself. How is this new framework going to completely change our understanding of life itself? It's a huge question. Keep asking those questions, keep exploring the data, and we will catch you in the next explainer.